So when we're looking at the gene that we try to transcribe, remember we have the beginning of the gene and the end of the gene. So something that students often confuse is start codons and stop codons, which are part of the RNA molecule. Sometimes students confuse those with the promoter and the terminator, which are part of the DNA. So the promoter is in the DNA. This is part of the DNA. And this is basically what says this is the start of the gene. So the promoter is where the RNA polymerase is going to join with the DNA and it'll start transcribing the message into an RNA message. At the end of the gene, there's another little segment called the terminator. The terminator is what tells the RNA polymerase this is the end of the gene. So that's where transcription will stop. So promoters and terminators, these are part of the gene. Promoters and terminators are the beginning of the gene and the end of the gene. And then everything in between the promoter and the terminator, that's going to be transcribed into an mRNA message. The start codon and the stop codon, these are parts of the RNA molecule, the messenger RNA molecule. These tell the ribosome this is the start codon. This is where we're going to start translating amino acids, and this is where we're going to stop translating amino acids. So promoters and terminators, start codons and stop codons, they have somewhat similar ideas, but they're used on totally different molecules. So let's take a look at what's happening with the RNA polymerase. The RNA polymerase joins the DNA at the promoter, and then it's going to travel along the gene until it reaches the terminator. And while it's traveling along the gene, it's going to start making an mRNA message. And remember what we talked about before, the mRNA molecule has the exact same message as the gene. It's just written in a different language. Now it's written in the language of RNA nucleotides. All of the RNA codons are complementary to the DNA codons, and we use the base pairing rule to determine which nucleotides are going to pair up with the DNA nucleotides. When this process is complete, we end up with an RNA molecule that carries the exact same message as the original gene, but now it's written in a language that the ribosome can read. So when the RNA molecule goes to the ribosome and the tRNA molecules will help to build the amino acid string or the polypeptide. So remember, mRNA, the job of the mRNA is just carrying the genetic message from the DNA to the ribosome and the tRNA. So again, we start with some DNA. This has the information for how to make a polypeptide. An enzyme called RNA polymerase will join at the promoter. It will read the DNA until it reaches the terminator. And while it's reading the DNA, it's going to convert the DNA message into an mRNA message. And the mRNA message will have the exact same information, but written in a different language. And the language that it's written in is RNA. Okay, it's written in an RNA language. Each letter of the mRNA is complementary to the nucleotides in the DNA. When this process is complete, the mRNA is going to leave the nucleus and it's going to go find a ribosome in the cytoplasm.